I'm very excited to introduce some of our speakers. These are people that work in the Center for Environmental Health Science Research. They're investigators, so they work at the University of Iowa in various capacities, and they do research on topics related to environmental health. And so this is an opportunity for you guys to practice some of those skills we were talking about in the panel of really listening to some hard science information, ingesting it, ask questions, and really try to understand, and they're going to communicate what they think are some important environmental health topics. So we're starting out today with Dr. Peter Thorne. He is the director of the EHSRC and is a distinguished researcher. So he'll be talking a little bit about his experience in the Science Advisory Board for the federal government. And then we have Dr. Robert Blount. He is a medical doctor who's focusing on tuberculosis. And then Darrin Thompson, he is a PhD candidate in the topic of occupational environmental health, and he focuses on neonics, and that is a really important issue, especially for bees and the health of our ecosystem. So, Dr. Thorne.
All right, well, thank you, Jackie. So I decided to give both a lamentation and a tale of moving out of one's comfort zone rather than give a science talk. And this stems out of my experience as working on the Science Advisory Board for the EPA for 6 years and serving as the chair of that Science Advisory Board. This is the principal board that advises the EPA, which is a scientific agency. And the board is created under 2 federal laws that were designed by Congress to ensure that the EPA gets the value of the very best science available in all of their decision making. So we live in interesting times, and this is a tale of that. And I guess the cartoon says kind of where I am with where we are now in terms of the value of science in the political spectrum. So the mission statement when I started working for the EPA is on the top here, and the current mission statement is down there. I guess we've dropped the part about air, water, and land upon which life depends. That was too controversial for today's EPA. So I served initially under Gina McCarthy, who was the EPA administrator appointed by President Obama. So she served, as shown here, until the day before inauguration, January 2017. She was a distinguished scientist, scholar, she still is, and her feeling was that the EPA is fundamentally a science-based agency and that rigorous science informs good environmental policy. When she was in the room, she was the smartest person in the room among the board of distinguished experts. She met with the Science Advisory Board regularly, and she would come and spend an hour and a half, which is really a pretty amazing amount of time for someone with a plate as full as hers. The next person I served under was Scott Pruitt. I never met Mr. Pruitt because he never came to meet with his Science Advisory Board. And is in his tenure as the administrator of the EPA, he pursued an aggressive, I would say, anti-science agenda. And of course, we know about some of the other challenges that he confronted in that role. And so some of them are here, and I don't need to talk about these, but it really served to undermine the morale within the agency. The EPA is an agency with some 18,000 employees nationwide, many of whom are scientists and who have dedicated their lives to environmental protection and the health of the environment. And so it was very disconcerting, and there was a lot of people who were choosing to do other things rather than remain employed with an agency to which they had devoted their whole careers. So just one example of some of the things that were coming out of the EPA suddenly was a refusal to understand the linkage between carbon dioxide and global warming. I mean, in the climate change debate, you can argue about how fast things are happening, to what extent it's human activity, how we can adapt, whether we should adapt or mitigate, what it all means, where do we put the resources to combat it. But to just deny at the very basic that CO2 increases and greenhouse gas emissions are contributing to global warming is a pretty extreme view. And then there was this statement how we've actually been on a hiatus since the 1980s, which is what he said. And this is the evidence. I think it's pretty clear that there's no hiatus in the increase in temperature globally with emissions. So this kind of thing coming out of the leader of an agency that is responsible for responding to greenhouse gas emissions, as was decided by the Supreme Court on 3 separate occasions, was disconcerting. And is this new science? Well, no. Svante Arrhenius in 1896 published on this and recognized that increasing CO2 increased the average temperature and could predict how much that would be, and he's the one who identified this as the greenhouse effect. And he thought, by the way, he thought this would be good for Sweden because it was too cold in Sweden. But this is not new science, and so to debate science that's been established for over 125 years really speaks to a disdain for science and scientists. So EPA Science Advisory Board was chartered under this law called IRTA, and it is to ensure that, as I said, the EPA gets the best peer-reviewed science in all its decision-making. And so the Science Advisory Board deals with very, very complex issues where the EPA is trying to make policy. They want to make sure they get the science right. And so the Science Advisory Board dealt with things under my watch: connectivity of waters of the USA, impacts of fracking on drinking water supplies, chemical risk assessments for very important industrial chemicals, carbon accounting for biofuels as we start to use agricultural residues and wood for as fuels to replace coal. These are complex issues that have political ramifications. They cost people money. They save lives. And so they're always difficult, and you need the best science and the best independent science in order to advise the EPA and make sure they do the right thing. This kind of advice was not valued under the new administration under which I served. This is what the Science Advisory Board looked like on the left before the inauguration. The members with no industry ties are shown in the blue. Those who have industry ties are shown in the pink, and then those with ties to fossil fuel, chemical, and tobacco industry are shown in the deep red. So you can see with the first round of people finishing their terms on the Science Advisory Board, new people coming on, there was a very dramatic shift in what the board looked like. This had never happened before under any Republican or any Democratic administration since the founding of the EPA and the EPA Science Advisory Board. So
there are no academics on the board anymore?
There were some academics, yeah. Here, these are many of them are academics still, and this is published from here, the citation, whereas I made the one on the left because I knew every person who served under my watch. Yeah, but the character of the board dramatically changed, as you can readily see from this. And so Gina McCarthy, in responding to this when she was giving a talk at Harvard after no longer being the administrator, encouraged scientists to become political. Scientists, by their nature, are sort of risk-averse, introverted, really don't want to go become advocates. And so she said that we really need to get into the political fray because these events have made our world become political. And so as a public citizen who is well informed, it's our duty to say something about this and try to act. So a number of us took this to heart, and 4 of the prior Science Advisory Board chairs got together and did some things. We wrote some op-eds, we did some media appearances, mostly to make the point that this is happening, B, it's not the way to operate a science-based agency, and see to try and put sunshine to this change that was occurring. So along the way, I was interviewed by Scott Simon on a Saturday morning. That certainly took me out of my comfort zone. And what was interesting is we're talking about pie shakes at the Hamburg Inn, and then he says, and 3, 2, 1. And then he asked me this riveting question that was a real zinger. So I had to kind of be prepared for that. So my comment in response to one of his questions was shown here. If Mr. Pruitt wants to form a regulated industry advisory board comprised of representatives from the affected industries, he can certainly do that, but he shouldn't expect unbiased evaluation of the science and policy implications from such a board. And then I subsequently was interviewed by Ben Kiefer on River to River. And we talked about this because a number of the people on the Science Advisory Board were finding out that they had been replaced when reporters called them and told them so, rather than being told by the agency or through the normal means that they had been replaced and their terms on the board had been terminated. So then myself and Deb Schwackhammer, one of the other former chairs of the Science Advisory Board, had an op-ed in the Register, and where we talked about what's at stake. I mean, what does this mean to the everyday person? We tried to get on that attacking on the scientific integrity is not only disheartening, but will result in long-lasting damages to our society, diminishing public health protections that we rely on, and denigrating the science that is critically important to this federal agency. And then we went on to try and give some examples of what this means in terms of clean water, clean air, clean soil. Again, this is something that I'm not trained for by, I'm usually a lung guy who's in the lab studying environmental lung disease rather than figuring out how to communicate these things effectively. It's easier in the print media than it is on radio or television. We've also been working with the new administrator. We write a 3-page letter. We get back a 2-sentence reply from a person 3 tiers down from the office. The other thing that's been happening is to scrub any mention of climate or climate change from the EPA's website. Again, this is an agency that's charged by Congress and with overseeing research and protections for climate change in the environment. So to have no mention of that on their website seems to be rather extreme. So before all that disappeared, we downloaded virtually all of the content, and as did a number of others, so it's still available elsewhere, but it should be on the EPA's website rather than having us have to host that material. So the status of environmental science in the U.S., I think the special interests are winning. Now the government is not helping us uncover the truth, but they're covering it with whatever that is that's being shoveled on top of it on the left. And I feel like we've really lost the special interest despite our best efforts to do otherwise. And I hope that things can turn around, and maybe even before 2 years from now. But this is a serious issue, and I think we need to, as scientists, be okay with stepping out of our comfort zones and taking on some of these big issues that are at the interface between our science and the politics of the day. And with that, I'll stop, and I guess I get a couple questions. Yeah.
I was just going to, Chris Martin from University of Northern Iowa. I was just going to ask, where would one find the EPA information that you copied from the site before they took down the information on global warming?
ehsrc.org.
Okay.
And we just redid the website, and Jackie...
They will be up tomorrow. We just redid the website, and I have not loaded the 52 fact sheets, but we have them all.
Yeah, thanks. Yes.
My name is Anna Spurry. I'm with the Des Moines Register. So when you, as a scientist, become political and start being more open about your concerns, are you at risk of losing funding at any point or not being accepted into certain grants? Is that something you worry about?
So So I have never been supported by the EPA. I've been supported by the National Institutes of Health, which is a totally separate process. A number of my colleagues who were demoted from various positions and who did have EPA funding did the same kind of things as me, making statements. In one case, one of my colleagues decided to go a legal path and sued the EPA over her dismissal from the advisory board. And there are certainly groups who are watching to see if there would be any retaliation, and she has not yet tried to reapply for new funding, so we don't know. But it is, of course, a concern that many people would have if their principal research is funded through EPA, and if their contract with their university requires that some of their salary be paid by federal dollars and research grants. So in my case, half of my salary must come from grants and contracts. So it is a serious form of intimidation that can occur. So that's a good question. Yeah, Emery.
The government's anti-science attack represents a fair portion of the public who's also has that attitude. I just wonder, from a science point of view, are you seeing that immunity to the facts?
So, yeah, that's an excellent question, Emery. I think one thing we've been seeing is it's kind of wrapped up with the fake news stories. You know, the whole notion that there's so much stuff out on the internet that's garbage or outright distortion. And we talk about that in one of the courses I teach on climate change. We actually look at stuff on the web that's completely fabricated, that has NASA's logo on it or the IPCC's logo, but it's really not. And I think what encourages me is that many school teachers, librarians in particular, are teaching young students and children to understand data sources of information, what's reliable, what is not reliable. And I think in the process of that, we might increase the science literacy and the value that people hold in credible science. That's my hope. But I think right now we're kind of at a low point in terms of the value that our society has for science. I spent a fair amount of time in Europe, and I think there's a different story there. And perhaps that's tied to the aggressive nature of Europeans' response to the climate change issue of the day, and the response that those countries are mounting to try and mitigate and adapt.
I think we're going to move on, but we'll have time for more questions later. Thank you so much.
Thank you, Peter. That was a great talk, and I sure do hope that you and other excellent scientists make their way back to the EPA board sooner than later. And Emery, to your concern about science and the public not wanting to not having appreciation for that. I'm a medical doctor, and I certainly see that there is an important intersect with health, because I do feel that my patients, the patients I see in clinic, are very interested in science. They're interested in their science and what's happening to their health. And I think are most of you journalists? That's great. I think I'm just putting in a plug for that if you can connect with people about their health, you may be able to get some interest there. I'm going to talk about air pollution. And in Iowa, we don't necessarily think about air pollution as a number one problem. I just want to say that in the United States, it has been a very big problem, and that government regulatory agencies such as the EPA have had a really important impact on air pollution over the since the 70s. When the Clean Air Act was envisioned and legislated in the early 70s, we've had a remarkable improvement in air quality in the United States. And a lot of us attribute that to our government regulatory agencies. I used to work in California. I was in the Bay Area up until about a year ago, and my kid was in soccer, and his coach, now in his 60s, was saying, I played soccer in Los Angeles when I was a kid, and I could hardly breathe. I was wheezing so hard, my eyes were watering, and I said, well, did they call off practice? They said, no, it was like that every day. And if you've been in LA recently, things have really cleaned up, and due to emissions controls, and that have been mandated by EPA and the California Air Resources Board. So a lot of good things coming out from those regulatory agencies. My focus is air pollution. We don't have an overall global problem in the state of Iowa, but we do have those pockets of problems, and my patients are telling me about that. I'm a lung doctor, and I see asthmatic patients in outpatient clinic, and they're saying, I live next to this factory on the Mississippi River, and the emissions are terrible. I really need help. What can I do? And so these are stories that we really need to listen to. My focus as a lung doctor is also on infections of the lungs, and I'm going to talk about one infection called tuberculosis. It's not a super huge problem in Iowa, but it is globally a big problem. And I just want to give you a little bit of science behind what is tuberculosis. It's caused by a small rod-shaped bacteria that people breathe into their lungs. And here you see a sputum with Mycobacterium tuberculosis in the sputa. The little red rod-shaped organisms are the Mycobacterium. It's spread very easily through the cough, the aerosol in the cough, from person to person. Clinically, it infects the lungs, causing fever, weight loss, and cough. Ultimately can spread throughout the body. It causes this wasting, slow wasting disease that before antibiotics we called this consumption. The tricky thing about TB is that you don't necessarily notice symptoms right away. So people can be infective, cough, and spread the infection. So that's one of the reasons that it persists after millennia. Risk factors: close contact, poverty, impaired immunity. And in my research, I'm looking at air pollution as a risk factor. So worldwide, there are 10 million cases of tuberculosis diagnosed annually, and 1 million deaths secondary to TB. It's now the #1 killer, infectious disease killer worldwide, even above HIV. So still a big problem worldwide. The incidence, you can tell, really varies from area to area. So you have big problems in Southeast Asia, over here, in China, India, and in Africa. So just one slide on air pollution, the science behind air pollution. Type, sources, and global distribution. So we can think about air pollution in some different ways, and an easy way to categorize air pollution is by physical state: gaseous or liquid or solid particle. And the gases, of course, ozone, sulfur dioxide, nitrogen dioxide. And then the particles, the particle I'm focusing on in my research is fine particulate matter of less than 2.5 microns in diameter. So that's about the same size as a bacterium. The reason that we're interested, and a lot of researchers are so interested in fine particulate matter, is that it's respirable deep into the lungs. And from that alveolar space deep in the lungs, it can be absorbed into the bloodstream. stream. So you have a pollution that is very bioavailable in the human and can cause organ damage throughout. The main sources, and my focus is on tuberculosis in Vietnam and Southeast Asia. The main sources of pollution there are from vehicles, and in the first picture down there you see a crowded street in Hanoi, the capital city of Vietnam, and it's crowded with small motorcycles, poor emissions. The stuff coming out of the small moped is worse than the emissions coming out of a large new vehicle in the United States. And you have literally millions of these bikes on the roads every day. The other big problem with emissions is coal burning for electricity. Even cooking inside the homes is happening with coal, and also heating. You can see the global distribution of particulate matter. The red, of course, here is high levels of particulate matter, the highest in the world here in India, also in China, and Vietnam is in Southeast Asia right down there. So my research question, okay, so air pollution in Vietnam is very bad. It's right up there with Beijing, with Shanghai. It doesn't get as much press. Tuberculosis is really bad in Vietnam as well. So I have some questions. I said, is there an association between the two? Does air pollution increase the risk for tuberculosis? And does air pollution somehow impair the immune response against tuberculosis? What is the mechanism there? And so 2 studies hypotheses. 1, fine particulate air pollution increases the risk for TB infection. And secondly, PM2.5 impairs airway epithelial cell killing of bacteria. I want to see if there's risk there and then see perhaps what mechanism might be driving that. And so we have recently set up a study in Vietnam, looking at that. This is a large prospective cohort study of 350 healthy children. Each of those kids have been exposed in the house to somebody with TB, and then we follow those kids prospectively over 1 year, quarterly, every 3 months, getting in touch with the child, measuring their air pollution exposures, and testing them for tuberculosis. Here we have just a schematic. We're collecting air pollution on Teflon filters, and you have a scanning electron microscope picture here with a small particle of air pollution on the filter fabric. Then we get the particles off into solution, and then we expose those particles to airway epithelial cells. So those cells in the airway that can actually kill the TB right then and there. Our hypothesis is that the particles are going to impede that killing. Our next steps after we enroll those children, we're going to we'll perform the study procedures, analyze the data, and report the findings. Importantly, I just wanted to say this in closing, and you know, it's hard to do this research. It's time-consuming, and I ask myself, why am I doing this? You know, what's the reason? And I think I have a good idea that there probably is an increased risk of this heavy air pollution with tuberculosis, and I want to get that word out. And so if we do find that there is increased risk of air pollution with tuberculosis, I think that's a message that really needs to be conveyed to people in Vietnam. And you know, people always ask me, so you do this research, you find something, you know, you find increased risk, so what? You know, the government's not going to do anything. But I think if people get this message and then put pressure on governments to make a change. Because tuberculosis is a really, we don't feel it here, but in Vietnam it's a scourge. It's like killing people. You know, 1/10 of people that get TB die from it. And so every single family has TB in their family. So that's, if we get this across, that air pollution is helping to drive the epidemic, I think we can see some change there. And I'll take a couple questions.
Thank you. Another question.
In your slide about risk, you had risk factors of close contact with someone with TB, impaired immunity, and poverty.
Yes.
Can you talk a little bit more about why poverty belongs in that list, or whether,
Yeah. Poverty is a big risk factor for TB, and I think one of the key features there is probably congregate living. Tuberculosis is directly the disease is the transmission is so important for that disease. And it's been the ongoing challenge of trying to knock out the transmission. And if you have a ton of people living in close settings, even in the household, right? If you have a lot of people in a small space with not a lot of windows, then you're going to really increase your transmission. We decrease transmission by windows because the UV light kills the bug, as well as keeping the windows open will help ventilate the bacteria out. And people in poverty, little hut, no windows. I think so that congregate, poor living conditions, and then malnutrition is another really big one that from that suppresses the immune system, the body's ability to fight off the infection.
Sure. I had a couple questions about the 1-year monitoring period,
Yes.
how you arrived at that with the quarterly checks, and then at the conclusion of this study, is this the basis for a larger study?
Right. So the 1-year checks, we found from prior literature that kids that are exposed to somebody in the household with TB, some of them do not go on to develop TB right away. In fact, it can take months, but within the first year after having a contact with somebody with TB, most of the kids that are going to develop tuberculosis do so in that first year. And then your other question was?
Would this study be the basis of a larger study?
Yes, definitely. So this is a relatively small study. It's going to be hard to make any definitive statements based on this. So it's kind of like a big pilot study. It's a little bit more than a pilot study, but we'll definitely need to do some ongoing work. Yes. What's that? Oh, yeah.
Hey, Zachary Smith, Press Citizen. I'm kind of curious about how you guys go about sort of measuring finding a child that has been exposed to one family member or more with TB. Like, how do you go about articulating that? Multiple exposure. I can imagine like multiple people exposed could complicate that. I mean, how do you guys go about wrestling with that variable? It sounds.
How do we identify those?
Identification and just it seems like kind of a hard thing to just sort of point to, right?
Right. And so we're operating this study out of 8 district health centers in Hanoi. So each of those district health centers is a TB clinic, and so each of the TB doctors is recruiting and enrolling for us. And so each doctor identifies the index cases, so patients with active tuberculosis, and those patients are going into then a treatment trial that somebody else is doing. And then my focus then is so we have the patient, then we also ask the patient, do you have any kids in the home? And that's where we identify the kids. And yeah.
One last question, and then we'll move on.
Hi, Lana with the Northwest Iowa Review. I'm curious, your study subjects, did they have access to the BCG vaccine, or is that was that not prevalent there?
Yes, everybody gets BCG, and the reason we do BCG in Vietnam and in most highly endemic countries is that BCG does work in young children, so it helps to prevent the severe manifestations of tuberculosis in 0 to 2-year-olds, and so that's why we're still using it in those parts. And we're actively working on a new vaccine that will help in people older than that, in the majority of the population. It's complicating because BCG can give a false positivity for our TB test, which is essentially the skin test. But that false positivity wanes over time.
Great.
Yep.
Thank you so
Thanks.
much, Robert. And I just wanted to point out that we will have these people here for lunch, so you can ask them any questions during lunch. So give them a hand.
Okay, so again, my name is Darrin Thompson. I'm a PhD fellow at the University of Iowa. I'm an epidemiologist. So my interest in neonicotinoid insecticides is primarily as it relates to human exposure and the potential kind of long-term implications of being exposed to these insecticides over a short period of time or a long period of time. And so my research really kind of is digging into that. So neonics are a relatively new insecticide compared to many of the other pesticides that are in use. So historically speaking, nicotine has been used as an insecticide, going back 1,000 years, even in some societies today. So I lived in the South Pacific, and on their crops they'd grow tobacco. They'd take the tobacco leaf, they'd grind it up, and they'd use the extract as an insecticide. The challenge with nicotine is it's not very stable in the sunlight, so it breaks down fairly readily. But because of those insecticidal properties, and considering where we've come from with some of the insecticides that have been used historically that had very pronounced health effects when being exposed to them, the industry kind of came back to nicotine in the 80s and designed neonic as a potential new insecticide that would be more stable in the environment, but still be able to have kind of that benefit of protecting crops when applied at low concentrations. So 1994, the first neonic, there's about, I think, 12 to 13 different types of neonicotinoids currently in use. The first one, called imidacloprid, was introduced in 1994, and over the past couple of decades, they've gone from being introduced to the most widely used class of insecticides in the world. They're one of the reasons for their increased popularity is that they're used not just for agricultural purposes, but they're used for in urban environments as well. They can be formulated in many different ways. So in Iowa, they're commonly used as seed treatments, and so we have a picture of that here. So essentially they're affixed to, say, a corn or soybean, and then as the plant grows, because they're highly water soluble, they're absorbed by the plant and become part of the plant. So where traditionally you'd take the insecticide, you'd spray it, when you take out the crop, you can wash it off. With the neonic, especially the seed treatment, you can't do that because it's become, say, for a corn plant, it's become part of the plant, so it's in the kernel. It also used flea and tick collars. They're used to treat emerald ash borer, used in many, many different fashions. Because they are applied at such low concentrations, and because of the way they're designed, so they're designed with on the structure of the neonicotinoid, there's a negative charge, and so the science behind that is that insects, mammals, humans, we all have what we call nicotinic acetylcholine receptors. There's variances depending on the organism, obviously, and how they're structured. Well, that negative charge is believed to impart kind of selectivity for insects, so that they have a greater affinity to bind for those insect receptors than, say, mammals. So in that way, they're able to be applied at low concentrations with the hope that they're going to affect the target insect that's being focused on without having a negative burden on humans or other mammals that may be exposed. Previous studies kind of globally have found that because they're so highly water soluble, they make their way into surface water and groundwater quite readily. So in Minnesota, Wisconsin, and from studies around the world, they've been found in surface waters and wetlands, groundwater. It's only more recently that we've started to look at these in Iowa. And a few of the studies that have been conducted, they've found it in surface water, they've found it in wetlands. And within the past few years, the EPA, Health Canada, and the European Union have started to look at possible health implications. And there has been very limited research done in that area, and that's kind of why I became interested in this topic. So these are compounds that are being applied everywhere. We're coming in contact with them in our day-to-day environment. What that means and how we may be exposed to it, I really felt needed to be explored further. So the way these insecticides work is essentially they bind to the nerve receptor. They bind to it permanently so that the signal that is being transmitted continues to be transmitted, and then ultimately the insect becomes paralyzed and dies. What has been found in some studies is that these neonics may have negative effects on non-target species, such as bees, monarch butterflies, even some animals such as birds that rely on insects. So there's been studies in Europe where they've looked at declines in bird populations, and they've attributed that to the insecticide killing off essentially their food supply. Some of the in animal studies, they found that neonics, especially in rats, can cross the placenta blood-brain barrier and can alter the neuronal activity in rats. And more so, some of the degradates of the more common neonicotinoids, when they break down, they lose that negative charge and become positively charged. And some of those degradates are actually more toxic than the original parent compound. In terms of human research, so I did a literature review looking at all of the data available. And primarily anything that looked at kind of mammal exposure came from lab studies looking at rats, goats, and animals. There wasn't much done in terms of kind of linking that to what this may mean for human health. I think there was a study where they also did a literature review, and they found 8 essentially publications, and that was published in 2017. 4 of those looked at chronic exposure. 4 of them looked at acute exposure, and those acute exposures primarily were individuals who'd consumed a large amount of the insecticide with the intention of committing suicide. The 4 chronic exposures were really based upon essentially secondary data sources, primarily from California. So California has a very comprehensive pesticide database, so knowing where pesticides are being applied in certain fields within the state, and then looking at birth defects registry and kind of trying to correlate, find associations between different cases in an area and pesticides that were being applied in those areas. In 3 of those studies, I think they found very weak associations with, I think one was autism, one was with anencephaly, which is essentially a birth defect that affects the development of the skull. Weak associations, but again, no real discussion on how exposure may have occurred and at what levels. So as an epidemiologist, the dose of the exposure, the magnitude of exposures is very important. So just because you're able to statistically associate something where there's an actual plausible explanation for how someone may be exposed to that compound is very important to figure out. And so that's what I'm attempting to do. So this is a map of imidacloprid, which is the earliest neonic that was introduced, but it's also one of the most common. So just between 1994 and 2014, you can see that its use has become quite ubiquitous. There's a couple other compounds, one called clothianidine, another one called thiamethoxam, which are used most commonly in the seed treatments, especially for corn and soybeans. And those maps look very similar. The difference being that those were introduced about a decade later, so in early 2000s. And if you look at many other pesticides, maps are very, very similar. In Iowa, again, really since 2003, 2004, about the time that clothianidine and thiamethoxam were introduced, the growth and use has been pretty exponential. So right now we're approaching about 400,000 kilograms being applied a year. In the scheme of things, there's about 40 million pounds of pesticides applied in Iowa alone. 400,000 kilograms is relatively small, but given the newness, it's still a pretty rapid increase. So to date, I've been focusing primarily on exposure through drinking water, and particularly well water. So I've conducted 2 studies where the results haven't been published as of yet. One, we looked at alluvial wells. So alluvial wells are essentially typically shallow wells that have geology that means that there's a connection between surface water and well water. So there's kind of a readily access for contaminants from the surface to make it into the well water. And so I partnered with the U.S. Geological Survey, and we looked at 39 essentially monitoring wells in kind of northeastern Iowa, southeastern Minnesota. And what we found there is that in these monitoring wells, 71% had at least one neonicotinoid that we were able to detect, and around 36% had at least 2 neonicotinoids. And this sampling was done starting in July, ending in November 2017. So typically, the seeds are planted in April, May, and it's intended that these compounds break down within a few months. But what we found is that even as late as November, we are still finding essentially these compounds in well water. So then going from that, knowing that we could find them in shallow wells, wanted to look at municipal wells, different types of aquifers, different types of well depths, to see if this is something that can be found in kind of municipal well waters in general, or is it specific to these shallower wells that we were seeing earlier. So in fall 2017 to summer 2018, I ran 2 sets of samples, each with around 100 samples with well depths ranging from 2,000 feet to under 100 feet, different types of aquifers, so alluvial aquifers and other aquifers that are much deeper. And with that, we were still finding in fall, winter, around 38% with at least one neonicotinoid. And then later, when we went back in the summer, so a couple months after sampling, trying to see if if there may be higher concentrations just after planting, or is it pretty consistent? We found 43% with at least one neonic. And the concentrations that we are finding were very, very low. So typically when you think of like an EPA MCL, you're thinking parts per million or milligrams per liter. We are finding them in the nanograms per liter or parts per trillion. So if, for instance, if you were to think, because these are kind of based upon nicotine, the average cigarette has anywhere from between 2 to 20 milligrams of nicotine. So, there's 1 million nanograms in a milligram. So if 1 liter of water has 2 nanograms of a neonic, the amount of water you'd need to consume to get an equivalent exposure to 1 cigarette is probably more than you'd want to drink in a lifetime. But the fact that it's still there, and at least from preliminary indications, is that concentrations remain fairly stable throughout the year, you are being exposed to it, and what that acute chronic exposure may mean over a lifetime. So if you're cooking with it, you're drinking with it, you're showering with it, those are things that I'm hoping to kind of study further as this progresses. So I currently have 2 ongoing studies. One is called the Northeast Iowa Well Water Study, and we started. The funding started in July. The sampling started in early December, and so we're working with National Cancer Institute, Agricultural Health Study. Agricultural Health Study is a cohort study that has 60,000 Iowa farmers enrolled for the past 25, 30 years. And so we had visited 47 households where we collected water samples, urine samples, and then collected surveys on diet and farming practices. And so based upon this, we will look for a number of contaminants. We'll look at the dietary history, we'll look at the farming practices, we'll test the urine and the water to see if there's any correlation between various practices and potential exposure. Sampling for this has been completed, and the laboratory analysis is ongoing. I'm hoping within the next year we'll begin to share some results of that. Another study is a study where I'm specifically looking at donors at the University of Iowa's Maternal Fetal Tissue Bank. So this is essentially expecting mothers can opt into this program, and they'd provide biological samples that would then be used for research purposes. And so I've been provided access to urine samples for 350 women, and I'm currently testing those samples for neonicotinoids. My intention kind of with these last 2 studies is that on the basis of what we find there, we may be able to develop more long-term studies to really start to answer some of those questions about what exposure may mean in terms of longer-term health effects. Kind of the conclusions overall is that the 3 most common neonics are frequently detected in groundwater. They're found at concentrations under 10 nanograms per liter, and especially in shallow, unconfined wells, so wells that aren't protected by bedrock, and that exposure assessment's ongoing, and hopefully I'll have more information in the coming years on that. And this is just a list of the partners I've been working with on the various projects.
We have time for one question, and then you guys can talk to them during lunch. Anybody have one?
Hi, Nora Paul, Grinnell College. I'm not sure, I know you're not all the way done with your research yet, but do you have at least any ideas or maybe expectations on what you'll find in regards to who is most vulnerable to the effects of these neonics?
At this point, I'm kind of trying to go where the data leads me, trying to keep an open mind. We're primarily looking at farmers right now, and that was really just kind of the logistics of it, having access to the agricultural health study and their long history of health information, and the fact that they've partnered with some of the people I'm working with made it kind of an easy partnership. But I think in terms of exposure, there's been studies where they've looked at produce and they've found residues of these compounds in fruits and vegetables that you'd buy at a grocery store. Even as we're developing the method for identifying them in urine, we've had some challenges in that the volunteers who have provided samples, we were finding in it, and so we had to determine, is this potential lab contamination, or is this really something that, individuals who live in Iowa City and have nothing to do with farming, are they really being exposed to it, or is this completely something else that we're seeing on the machine that may not even be a neonicotinoid? So I think it's likely that most of us are being exposed to them at very low levels because they're so ubiquitous. But what that may mean and who may be most vulnerable, I think the data that we discover will kind of dictate where we go.
And Darrin, are fetuses and babies that have developing neurologic systems, are they going to be at increased risk? Yeah. This is a neurotoxin, right?
Again, this is just my opinion. The European Union did identify, again, a couple of these compounds as potentially neurotoxic, but again, that was based upon animal studies, and oftentimes the translation from what happens with the animal in the lab to what it means for human health is very difficult, and it may mean nothing. Just because you see a neurodevelopmental issue in a rat doesn't mean that it's going to necessarily translate to a human. But kind of personally, that was what drew me to this. So seeing that and thinking, what does this mean for a developing child? That's something I'd like to explore. And so that's kind of why I started it. But I've not seen any peer-reviewed published data to really give me a strong indication that that is occurring. But that again, just in terms of biomonitoring studies, there's been a couple in China, there's been a couple in Japan, and there's more recently a study that was published here in the U.S., but up until a few months ago, I'm not aware of anybody other than my group of researchers who was looking at it in the United States. So there's just so little information available to really say anything definitive.